For a successful PCR, the key component, the primers, their properties and their efficiency in amplification matters a lot. The points which should be considered while designing the oligonucleotides or primers are the base composition, length of the primer, the repeated sequences within the primer, the complementary nucleotides within the primers, melting temperature of the primer, and the 3' termini of the primers. GC contents of the primer should not be beyond 60%. An ideal percentage is 55%, but it can range from 40 to 60%. And there should be an even distribution of all 4 types of nucleotides while deciding to design a primer sequence. The ideal length for PCR, the primer should be 18 to 25 nucleotides long. We use a pair of primers. One is called as forward primer, the second one is called as reverse primer. Both of these primers should not differ from each other in length more than 3 base pairs. Like if the forward primer is having a length of 21 nucleotide, the maximum length the reverse primer can have is 24 nucleotides, or 3 nucleotides less than the 21 nucleotides. Keep in mind there should not be any repeated sequences, inverted repeated sequences, or self-complementary sequences in the primer. In such cases, the primers can achieve secondary structure and they can make the loops in their single-stranded structures. The 3' ends of the primers, they are very important because these are the points where the extension will occur to prepare the daughter strand. This terminal sequence of one primer should not be able to bind to any site of the other primer. The primer's concentration, if they are present at high concentration, there is likely a chance that the weak complementary bases, they can base pair and they can make the hybrids or they can make the dimers during amplification. Melting temperature is important for the primers. This is the temperature that separates the primers from the template DNA strand. Annealing temperature is a temperature at which the primers bind with the template strand. This temperature is calculated using an equation, and the 2 primers should not differ from each other in melting temperature more than 5 degrees centigrade. And when the amplification of the gene is achieved, you should pay attention that the melting temperature of the product should not be different from the melting temperature of the primers more than 10 degrees centigrade. Because when the second or third or subsequent cycle has to go and the denaturation step has to follow, a huge difference between melting temperature of the product and of the primer can cause problem with the PCR. The nature of 3' end is important. It is crucial. It is suggested that there should be a G or C nucleotide at the 3' end of the primer. However, C, G or GC together are not recommended to be present on 3' end of the primers.